on the surface of the red blood cell, there are a whole series of antigens. Well, one of them codes for this ABO system. And O-type ends abruptly at galactose, on top there. People with blood type A also have an additional sugar, acetylgalactosamine. Those with B blood type have another galactose antigen on the end. So O blood type is essentially the absence of one of these other sugars being added to it. And that is the reason why A is dominant over O, B is dominant over O, and A and B are co-dominant because both have this additional sugar. Recessive traits are usually the absence of something. That is the failure to produce something. If you look at the surface of the red blood cell in this sort of flaky picture, people with A blood type, forget the second line, we'll go over that later. On the surface of the red blood type, they've got the A sugar chain. B blood type has the B type of sugar chain. AB has both A and B sugar chain, and O has neither of those two dominant sugar chains. It just ends abruptly without adding that last sugar. Now, the A blood type is can be produced if you have two A alleles, homozygous, or an A and an O, because O is recessive. B blood type individuals are either homozygous for B or heterozygous for B and O. And if we could do, we can't do this anymore legally, but we used to do this in lab. And you can test for your blood type, and there's an anti-A compound, an anti-B, and this is what it looks like when you just have blood splop with something put on it. But if it's anti-A, this is the A blood type. This is what it would look like. You put anti-A on one drop, anti-B on the other. Anti-B does nothing. Anti-A will make the red blood cells clump. This is the B blood type. It does nothing to the A blood type, but it clumps the B. If you have AB blood type, both of these will clump. And if you have O, you just don't go anywhere. Nothing happens. Now, there are also subtypes of A, subtypes of B, et cetera, but these are the main points. Well, the second law that Mendel suggested was the law of independent assortment. And this deals with comparing two traits. Now, again, he had 7 traits that he tested for. Each one was on a different chromosome, and that's why he ended up with this. If 2 traits are on different chromosomes, the grandparental traits will not necessarily be inherited together, because there is a reshuffling during the process of meiosis. This, once again, is one of the miracles of sex, that you don't know which chromosome you got from which parent unless you can test back to the grandparent. So, if, in this case, if the ABO blood type, as in AA, is on one chromosome, and the MNSS blood type is on the other chromosome, and one individual, the dad had blood type AAMM, or had the genotypes AA and MM, and if mom had different ones, she had the genotypes EB and N, which is the other allele for the MNSS system, and they're on different chromosomes, you're not going to get dad's AM necessarily inherited together. They will be inherited together only by chance. And mom's the same. So you, once again, you get a reshuffling of the deck. So when meiosis occurs, chromosomes will separate out, not necessarily paternal alone on one side and maternal alone on the other. Well, we've been talking about genes and genotypes and phenotypes. Let's talk about the genes themselves. For many years, the question was, what is a gene? First of all, we didn't know what chemical it was. We figured it was a chemical. People fiddled around with it. Finally, two bad boys, Francis Crick and James Watson, decided to tackle the problem. That's Francis Crick on the left. He's British physicist, and James Watson on the right, and he was a bird ethologist with his dissertation. But Watson decided that a very interesting and important thing you might find out is, what is a gene biochemically? And so they fiddled around, and finally came up with the correct answer. That's, I should have it reversed there. It's Watson on the left and Crick on the right. And this is what time does to you. Time is terrible. Or at Christmas time. If you want to read how they got it, an extremely readable book, a bestseller called Double Helix, was written by James Watson. It describes from his vantage point how the whole process went. He created a whole series of really stupid screw-ups on the road to getting there. There were some really dumb things that happened, but they finally put it together. Anyway, the book cost Watson every friend he ever had because he was very blunt about what he thought. Instead of being a little suave, he just said what he thought. And the first sentence in the book is, "I have never seen Francis Crick in a modest mood." And that sort of started the whole thing off as losing every friend he ever had. Anyway, he talks about Linus Pauling, he talks about other people that were involved, that were British. Linus Pauling was from California. He was a character. It was an extremely readable book. It was a bestseller and scandalous. If you like a lot of gossip and political intrigue and backyard backstabbing, etc., it's a great book. What they found was a double helix. Finally figured it out. It was a double helix with a backbone on the outside, this swirly thing, ergo helix, and linking these two helices, one here and one here, together were a series of bases. And there were 4 bases only: adenine, thymine, guanine, and cytosine. It was a very simple setup. This is how genes work in humans, in chimpanzees, in fish, in broccoli, in bacteria. It's the same system. Notice the fact that there is a double helix. Notice also the fact that the illustration has adenine linked up with thymine and guanine with cytosine. In point of fact, these are obligate relationships. Adenine will only link up with thymine, and guanine will only link up with cytosine. So that if you know that you have guanine here, you know very well you have cytosine over here. You know you have thymine here, you know very well you've got adenine over here. These are obligate bonds.